Right, so this one is about DNA and mRNA or messenger RNA. More about that in a second. If you recall, the chromosomes in a cell are inside of the nucleus. Okay. Now this bit here, a bit beyond what we need to know, but this is called the nuclear membrane, and this is the bit that surrounds the nucleus in a cell. If you think of a cell like that, there's the nucleus, chromosomes inside. This is just a close-up of it, and it's actually got small holes in it, which will be important in a moment. The chromosomes normally are long, twisted structures like this, and all the way along this length of this chromosome there would be genes, sections of DNA that can be read off and used to as the code to make proteins, which is what will happen in a second. So we've got these long chromosomes, but they are too big to leave the nucleus. Now the problem we have is that the DNA, each section of DNA or each gene, gives you the code for one protein. And a protein could be an enzyme, it could be an antibody, it could be structural proteins like collagen found in your skin, keratin found in your nails. Each gene gives you the code for one protein. Remember, a gene is just a big section of DNA. Now, proteins are made out in the cytoplasm of the cell. So we've got the problem that the instructions are in here, inside the nucleus, but to make a protein, we've got to get the instructions out. And this is where the molecule RNA comes in. What RNA does, it forms in the nucleus, and it forms a copy, a template, if you like, of a particular section of DNA or a particular gene. And that RNA can then leave the nucleus, because it's a small molecule, and enter the cytoplasm. So it's known as messenger RNA. It's taking the message from the chromosome in here out into the cytoplasm. Now in the cytoplasm you have these little blobs called ribosomes. And what a ribosome does is the messenger RNA kind of sits inside of it like this, and the ribosome runs along and it reads, so it moves along, it reads this RNA sequence, and from this RNA sequence it will build a protein. So let's just look a little bit more detail at that. If you recall, the DNA structure is a twisted spiral or a double helix, a bit like a spiral staircase, or if you imagine a ladder that's been twisted round. So you've got two edges or two rungs to the ladder, and then between it we have these chemicals, these four bases as they're called, and they're given the letters A, C, T and G. Now you will have to remember that the sequence that these always line up in, T is always opposite A, C is always opposite G, so we continue filling that in. And these sequences can go on for hundreds and thousands of nucleotides, so we'll just fill a few more in just because. There we go. Now, when the DNA is still back in the nucleus, when the cell has worked out which gene it's going to use, a little section of that chromosome becomes available. It unzips. So if you like, one of these strands here becomes exposed. So perhaps the way to imagine it is, let's say we've exposed this strand of DNA, an RNA strand will come along and copy it. Now, RNA is a single strand, and there's a slight difference with it. There we go, there's our single strand. These chemicals, the T, the C, the A and the G, are floating around free in the nucleus, and what they will do is they will match up. However, in RNA, the one difference is that instead of a T, it's replaced by a U. It does exactly the same job, but it's replaced by U. So wherever we'd have a T in DNA, we would have a U on RNA. So if I fill these in now, so A would normally be opposite T, but it's an RNA molecule, so I'll put a U. Continue along here. Okay. So RNA, a single strand, and it has U instead of T. That RNA can then move off into the cytoplasm, where it can be used and read by these ribosomes in order to make a protein. Now, the last bit of this is that every 3 letters of this DNA code, so each 3 letters, gives you the code for a particular amino acid. Now, amino acids, there are 20 of them in your body, and if you think of them like building blocks, if you think of Lego bricks for a second, you could have a box with 20 different types of Lego brick, and from that you can build lots and lots of different models. So amino acids like individual Lego bricks, and they're used to build bigger models or bigger molecules, which are proteins. So amino acids are the building blocks for proteins. Each 3 letters, or each codon, as they're called, of the DNA code, gives you the code for one particular amino acid. So for example, if I just give you a few examples, you won't need to remember these. There is an amino acid called leucine. Usually they're shown by just writing the first 3 letters. The code for leucine would be CTT. We'll call valine, and the code for valine would be, there are several ones, that's GDA, for example. So these codes, each 3 of them, tells you to put in a particular amino acid. Back to this ribosome then, what it does is it assembles amino acids in the correct order, depending on what the code was along this RNA.